on way too many words for me to list here. And today's going to talk about analysis of cell-free DNA using a human methylation atlas.
Methylation. Yeah. You always say that about the words. Okay. So, fine. So, when I prepared the talk, I got scared that maybe you won't understand some of the things that I say. So just stop me and ask. That's the privilege of sitting in the lecture room, so don't hesitate. So remember the Tricorder. Maybe some of you are old enough to remember Star Trek 66. They had this device. This is not a cigarette lighter. It's something that you scan against a person or an alien, and suddenly you know what's like about all the diseases and their health and whatever. So we wanted to build something similar. Not as accurate, maybe. But the idea would be to draw blood of a patient and then to centrifuge it and to look for small pieces of broken DNA fragments in the plasma. Basically, those are pieces of DNA that come from cells that recently died. And those are called cell-free DNA, DNA out of the cell. Okay? And the idea is to then take those pieces of DNA and to be able to associate them with the tissue that they originated from, maybe from the liver or the brain or the pancreas or the lung. And then we would get a count of what was the rate of cell death in my body in different tissues in the past 15 minutes or so. And that would hopefully reflect my general health. So that's the goal of the talk. Okay?
What do you mean by centrifuge?
Centrifuge is centrifuga when you put a piece of DNA, a bottle of DNA, and you blood, sorry, and you centrifuge it, and then it separates to red blood cells and white blood cells and the liquid above it. Okay? So that's the idea. And let me, so the main player here, one of the two players, is this thing called circulating cell-free DNA. So those are very short pieces of DNA. They're about 150 bases long. They are released from dying cells. They're constantly flowing in our blood, and they're being cleared in the liver with a half-life of about 20 minutes or a quarter of an hour. So if I would be able to associate those with the tissues, I would really get a very online image of my health. Okay? And in 1 cc of plasma, which is about 2 cc of blood, which is like half a spoon, we have about 1,000 fragmented pieces from every position in the genome. Okay? And actually, you've heard about similar approaches. There's something called NIPT, which is non-invasive prenatal testing, which people are using to look for trisomy, Tismonid Down, for instance, in the early stages of pregnancy, and also liquid biopsy is the same approach that use those fragmented pieces of DNA in the blood to monitor and to identify cancer. But those two approaches rely on differences in sequence between the host and the foreign DNA, between the baby and the mother, or between the tumor and the normal DNA. Okay? So by estimating those differences in DNA, you can associate every fragment to the host or to the foreign origin. Okay? And we have a problem that all the cells in our body have exactly the same sequence of DNA. If we remember, we started from an oocyte meeting a sperm, and we had one copy of DNA in the oocyte and one copy of the DNA in the sperm, and together they make the embryo and then make us. So all the DNA is exactly the same in terms of sequence. Yep, but it's packaged differently. It's organized differently in the cell. You take 2 meters of DNA and you need to insert it into a 10 nanometer, which is like 6 orders of magnitude, into the nucleus of every cell. So you need to package it really, really condensely, and that's different in different cell types. And another thing that is different between cell types is the expression of genes. So here you see 3 different examples of genes that are expressed, and they're colored in blue. So this is one gene that is expressed in a mouse embryo in day 10, in the limbs, maybe in the nose, maybe in the brain. Another gene that is expressed only in the eye and heart and the spine of a chicken embryo. Another gene that is expressed in 7 different stripes along a fly, a fruit fly, embryo. Okay, so genes are expressed differently, even though that the DNA is exactly the same. And there's a whole set of mechanisms that control where the genes, in which cells the genes is expressed, and in which cells it is not expressed. Okay, and those processes involve the sequence of the DNA and the accessibility. Some regions are open and some regions are closed, and those two together affect the binding of proteins. Some proteins can attach to the DNA and then express the gene or not express the gene. And the packaging of DNA of chromatin, which is what Nir is focusing on. And then you also have the 3D compacting and the 3D organization of the DNA. So some regulatory regions that are afar could then fold over in specific cell types to activate faraway genes. All those are different in different cell types, except for the sequence. Questions? Yeah. Chromatin is the organization of DNA on those proteins that are called histones or nucleosomes. Basically, it's too long, so you need to hierarchically package it again and again and again. Yeah?
When you say you express genes in certain areas of the body, what do you mean exactly?
So you have the DNA, which is something that is written on the genome, and then you have RNA, which is like a more contemporary copy of this gene that you can make proteins of. Okay? So we want to use those features to identify the origins of cell-free DNA. And this is Nir here sitting in the back and folding origami with his group. And they published last month a beautiful paper about using differences in the packaging of DNA in histones to identify the origins of cell-free DNA. Okay? So that's a new method that they've been working on for some time now. But we want to take another approach, something that is called DNA methylation. So as computer scientists, this is like super cool, so you need to listen. The genome is about 3 billion bases long, okay? 3 to the 10 to the power of 9. And then about 1% of the position in the genome contain the sequence C and followed by G. Cytosine followed by guanine. And every time you have a CG, you basically have a bit container that the cells could use. So every CG in the genome could be either in the methylated or an unmethylated state. Okay? The cells do that by themselves. And this is a method for the cells to basically expose some of the regions of the genome while commenting out the rest of the genome. So most of it, it's exactly...
CG is not part of the genome.
Yeah, CG is part of the genome. You have the letters. They have the same meaning that you have, but you have another level of annotation that you can hide a bit here whether this CG is, the C is marked or not marked. It's just as the red sticker that you get in the airport to the back of your passport. It doesn't really do anything, but some people can read it and know what it means. So again, here we have this methylation bit. And if you have it, you basically comment out this region. So you make this region inactive. Okay? So this is a way for the cells to have the same sequence of DNA, but then to highlight different regions that are relevant to specific cell types. Yep? Okay, cool. So we have 20 million such sites, C followed by G sites. And you have, in principle, you would have 2 to the power of 28 million different patterns.
But each one is commented until the next CG, or?
No, you have this one, this would be commented by itself.
Then what is commented?
And then when the cell would divide, the 2 cells that would be the next generation would have the same, this mark would be maintained. And also, like, if you have cells that don't divide, like cells in the brain, we basically got the marks in the cells, in the DNA in our brains when we were about 10 days or 2 weeks, and the mark persists for the entire life.
No,
So
what is the scope of the comment?
no one knows. It's local, I think. A protein that would be here would see it.
Only there.
Yeah, yeah. So I'm afraid to answer what is this.
But it's not a start comment or end comment like we.
No, it's old style commenting like the hashtag.
One basis, one.
Yeah. So if you have a gene, and you have this region just upstream to the gene being marked by being methylated, so this gene would be inactive. because the start is marked as, is basically commented out, and then you won't express this gene. So there is a question of scope, but
for the whole gene, not for
yeah,
the base.
yeah.
So something in the middle, in the beginning
Yeah.
of the gene, there's something saying start
Right.
treating this as a gene, and you kill that.
Yeah.
And the next gene will have its own start treating.
Yes.
Now, it's more complicated. There's 28 million in there. It's not 28 million genes.
You have 23,000 gene and 28 million
So there's-
methylation, so you have 1,000 to, yeah. Okay. And something that we could do, we could measure either the average methylation of specific CPGs, or we could sequence a piece of the DNA. So the highlighted yellow region is something that we could sequence. We could read the sequence using a machine. Sequence is reading, not generating, but reading. So we could read this yellow portion, and then we could also infer in some method that I will not explain about whether this one is methylated or not, and this one is methylated or not, and this one is methylated or not, okay? And then we could basically consider this DNA methylation data just as a stick with 3 beads on it that could be either methylated, each one, or unmethylated. So we have 3 bits.
I mean, there are 3 possible places for methylation, let's say, in this whole yellow sequence all over
Yeah.
the world.
Yeah, we know where the CGs are
possible
in advance. Yeah. So if I have the start position for the yellow region, and I have 3 bits, I would know what are the indices of this one and this one and this one. It's very useful. Okay. So now let's do some linear algebra. So what we did some years ago was basically to take DNA and then to measure the methylation, the average methylation in, let's say, 500,000 positions in the genome. So you have a vector of 500,000 with average values in there, the average methylation of this site and this site and this site and so on. And we would want to understand what this blood sample or plasma sample contains. Basically, what cells died to give birth to those pieces of DNA that we see. Okay? And this is classic linear algebra, multiple linear regression model. You can basically say that this vector here, we could fit it using a combination, a weighted combination, of all the different types of methylation that we have for different cell types. Okay? So basically, you would say that this is composed of 10% of this column and 5% of that column, and so on. Okay? So this is basically using and if you're smarter, then you would, instead of using ordinary least squares, you would want to use non-negative least squares, because you can't have negative contribution of some cell types. But basically, using L2 norm and non-negative least squares regression model, you can now, and given an atlas which specifies how the cells are methylated in every site in the entire set of known cell types, we could then deconvolute, like the biologists like to say, this sample and estimate how much DNA we have from each type. Was that clear enough? No? Algebra linearit, you have a vector here, and then you want to express this vector as a combination of the columns. Each cell here is one site, and then you would have the average methylation of that exact site in all the different cell types. So every row would be an equation, and you want to solve the 500,000 equations together up to some noise and estimate the contributions. So for that, you would need an atlas. So together with Yuval Dor and Ruti Shemer and Ben Glazer from Hadassah, we set out to build such a human methylation atlas, and we did that some years ago. And we published this atlas with 25 different tissues in it. Some we collected from publications, and some they measured in the lab. This is the average methylation in 25 tissues, like kidney, even though that the kidney is composed of various cell types. But that would be the average methylation. So if you look here, and those are only the positions, the equations, that are differentially methylated. So for instance, here, we consider liver hepatocyte, which are the cells that make the liver. And up here, you have about 100 different places in the genome where cells from the liver are 100 or close to 100% methylated, but all other cells, in all other cell types, they're unmethylated. Okay? So that's like a useful equation, basically. If you look at here, down here, you see another set of about 100 positions in the genome that all the cells are methylated in those regions except for the liver. So they're unmethylated specifically in the liver and methylated elsewhere.
So this 6K is 28 million?
From the 28 million, we measured 500,000. And from those, we selected 7,000 positions, which are the most informative. Okay? Because you have noise in the data. So we saw that as we reduce the number, if we focus only on the useful equations, we get higher accuracy. So this is something that we can do then in the computers. We can basically do an in silico mixing experiment. So we just mix. It's not really, it's in the computer. Different percentages of liver cells into blood cells. And then we could estimate what is the predicted. We can use this regression model to infer how much DNA we put from the liver. And you can see, so this is the interesting range between 0 and 10%. And you can see that it works quite nicely for most cases. And we did that for the entire set of 25 cell types that we have. And you can see that all in all, they worked really nicely. The average error was about 1%, which is nice, but is not maybe enough for early detection of some diseases, but it's very useful for other diseases. And this was only done with 8,000 equations, which we tried to localize together. So we could measure some equations together. Yep. So all this was published. And then we used that. Oh, an important thing I want to say that the false positive. So when you actually mix 1% of something into the blood, we saw that the false positive rate was less than 1/4% on average. And the detection rate or the sensitivity was 70%. So it's very accurate at 1%.
Can you explain again how did you produce the prediction here in these graphs?
Yeah, so for every graph, for every percentage, you mix in the computer, you just do the algebra. Let's say 5% of bladder methylation with 95% of blood. And then you run the non-negative least square algorithm, and you get a combination. And then you look and you ask yourself, how much of the bladder the model is predicting to originate from the bladder? And then you plot it as a box plot, which is basically the 80% or 50% or whatever. You do that multiple times. Okay. So we did that, and then we turned out to test and to analyze plasma of healthy people. And what we saw kind of made sense. The DNA for healthy people is mostly composed of white blood cells, which are the cells that circulate in the blood. And then you have a portion which are megakaryocyte and erythrocyte progenitor cells. So those are the cells that make red blood cells and platelets, the COT, which is another known large contribution of the cells that we have in the blood. And then we had 1% of hepatocyte from the liver, which kind of makes sense because the liver is a big organ. It's dividing all the time, and it's floated with blood. So that made sense. And the rest are vascular endothelial cells, which are basically the interior coating of blood cells, or of blood vessels. So those are the pipes that, like you have rust in your drinking water, we would have DNA that come from the cells that make the blood vessels. So that's normal. And we tested, Josh Moss, the student who did that, went to elderly houses, and he tested older people and younger people. And we saw that in males and females, and it's more or less the contribution is the same, although older people tend to have a little more cell-free DNA because the liver is maybe not cleaning it as efficiently as the younger people. Yep. But then we went to see people that have diseases. So one nice case is something called pancreatic islet transplantation. Basically, you take pancreatic islets from cadavers, and you inject them into the livers of diabetic people. And then they produce insulin and they cure them out of their diabetes, basically. And we saw that in their blood, an hour after the injection, there's a huge fraction of pancreatic DNA which disappeared an hour later. Okay, so this is due to the injection, to the transplantation. Or if we looked at the plasma of a colorectal cancer patient, we saw that there's a huge fraction that was analyzed by the model or predicted by the model to originate from the stomach or from the rectum or from the colon. Okay? I remind you that the atlas is made of healthy tissues, and this patient had a cancer in the colon, but the DNA methylation of the tumor is similar enough to the tissue that it originated from. Yep? Sorry. Okay. So this was all published, and that was the old thing. And now we want to move to the next generation, and it's like a lame joke about next generation sequencing, which is how we measure DNA. So the idea would be different now. Instead of looking at the average methylation in specific positions, we would want to sequence binary patterns like fragments of DNA that could have multiple sites on them. So every stick with the lollipops here would mean that we have a sequence that starts here and have 3 adjacent methylation sites on the same molecule. So they came from the same cell.
There's a special way to measure average methylation?
Yeah, there's a cheap way to measure average methylation. Yeah, using an array, and this is using sequencing, so it's a different method. And we thought that now we should build a new atlas which would be larger and cleaner and more specific and could make use of those dependencies that we have between adjacent sites. Okay? So we were lucky enough to team with a company called Grail, which does early cancer detection. And they sponsored some of the research and mostly sponsored the sequencing cost of this new atlas. And it was it is a huge atlas. Basically, we now contain 28 million sites across the entire genome. Each position is covered on average by about 40 different molecules. So it's much richer in depth. And also, we sequenced 213 different cell types, or 213 healthy samples that are together make about 50 different cell types. So it's cleaner, it's richer, it's deeper, and it's wider. But it's not average, it's binary. And we basically tried to, together with Ben Glaser from Hadassah, he basically tried to contact all the physicians and all the surgeons that he knows, and we tried to gather whatever samples of DNA that we could get our hands on from healthy individuals. And you see that we try to focus on the blood and lung and breast and pancreas and the skin, all the places that we have diseases that we care about, and heart and brain, and the GI tract from the gastric to the colon, reproductive system, muscles, so on. Okay? So how do you analyze such data? So Netanel, who is here, hi, and Sapir, who is graduating tomorrow, wanted to analyze this data. And they thought that working on 213 samples across depth of 40 across 28 million sites is maybe too much. And we would want to segment the genome into blocks. Blocks are basically localized regions that have similar methylation pattern. Okay, so we would want to compress them and identify them. And then we would want to identify those that change between different tissues, and those would be the features that we care about. So you could think about that as feature extraction and feature selection, maybe. So Netanel wrote this something which is really cute as an exercise. It's basically a multi-channel, piecewise-constant segmentation algorithm. So you could do that using dynamic programming. So think that here you have 1,000,000th of the genome, and every dot represents the average methylation at a specific position. And you have the colon and the heart and so on, like 211 other different samples. And what you would basically want to do, and you would want to identify that here there's a stretch that in all the different cells, they look more or less have similar methylation levels on average. Okay, so it's like a piecewise constant. So we did this segmentation, And then we wanted to identify blocks or positions that differ between different cell types, that maybe would be methylated in the colon, but unmethylated in the heart and in all other cell types. So that would be a colon marker. Okay? Questions? You don't care anymore?
How many cell types do you work with?
50. But this is only, but I think he ran that on each sample together.
Do you only care about things that are one against everything else, or any binary?
So here we had any type that shows some differential methylation. Later on, we would only focus on those that are one against everything else, because they're easier to interpret. So once he did that and he tossed away all the data except for those 150,000 regions that are blocks and show some variability, so this is an unsupervised clustering of the different 213 samples, and the labels are colored based on the true identity of each sample. And you can see that it's continuous. It's really easy to see that the clustering is almost perfect, because you have all the pancreatic beta cells together, and then the pancreatic alpha cells, and then the delta cells. And so this is one tree. And then you have the pancreatic duct and acinar and the liver hepatocyte, which makes sense that they cluster together in a different subtree, but then they merge together and so on. So basically, this recapitulated more or less the entire richness and structure that we have in human DNA or in human cell types. Okay, so it was very cool. And then we wanted to come up with the next goal, which is basically finding statistical model for inference. So now we have those features, let's do some science with that and identify how much cells are dying from each cell type. And this is Sapir's master. So let me show you just one example, which is, again, a millionth of the genome. Here you have 1,500 bases, 35 methylation sites. Here you see the average methylation pattern, so every row is a sample. And here you see the actual data. So for instance, this stick here is one molecule, one read that we sequenced with 7 consecutive sites that are all unmethylated. And here you have another piece of DNA with 2 sites methylated and 4 unmethylated and so on. Okay, so you understand this, right? And as you can see, so this is one sample from the lung epithelial cells, and this is one sample from non-epithelial cells from the lung. For instance, the muscle cells from the lung. And you can see the difference. You can basically see that the reads in this region here in the lung epithelial cells are unmethylated, but they're fully methylated in the lung, in other lung cells, and basically in any other cells in the body. Yep. Okay.
What does each row, does it represent?
So we're just stacking them together. So whenever you have a free space, you could put them. The rows don't mean anything here. Just the strings or the fragments. Every fragment is a fragment of DNA that we sequenced. Okay? So basically, if I see one of those green guys, I would know for sure that they originate from lung epithelial cells, because in all the other cell types that we have in the atlas, it's completely methylated. Okay? And so this is this block here. And right next to it, almost immediately adjacent, we have another block who's differentially methylated in different cell types. Here we have this region that is unmethylated in oligodendrocyte, which is a type of cells that we find in the brain, but is methylated in the neurons and in all other cell types that we have in the atlas. So now if I see a green guy from this region here, I would know that it's from oligodendrocyte and not from other cell types. So you got the idea? So basically we have those regions that are so differentially methylated that we could use those methylation patterns to identify them in the blood and to count them as recent cell death events from those specific cell types. Yeah?
You said earlier that there are about 50 sites, right? Lung.
Here, in this genomic region, we have 35, you mean cell types?
Yeah.
So here I'm showing just some of the cells. We have more. But you need to trust me that all the other cell types would be completely methylated in this region. Okay.
What about differences between individuals?
With individuals. So we wouldn't know. We think, okay, so the first answer should be that methylation patterns between individuals are very conserved. It's only when things go wrong, like in the cancer, each cancer cell or each tumor in different people would have like a different trajectory. But as a rule of thumb, the methylation is very, very conserved between different individuals. Yeah, you can take the platform that we used before with the average methylation.
If you take 2 random people, healthy people,
It would be identical. If you would look at the average methylation at specific cell, at specific position, and you would have they would deviate by 2%, 3%, which is about the measurement noise of the array. Okay. So now imagine that we have DNA from the blood here, like the entire pool of DNA. Each fragment is from a different origin. But then if we see something that is completely unmethylated in this region, which is only unmethylated in those lung epithelial cells, or if we see an unmethylated fragment in this region, which is only unmethylated in the oligodendrocyte cells, so those would be witnesses of a cell death event in those specific cell types. Okay, so this is what we want to do next. So there's a whole lot of things involved in here. We basically want to select those windows, and we want to measure the likelihood, I will show it in a second, but to measure the likelihood of each signature or each pattern. And we want then to use that as a classifier and to learn a classifier and then to analyze the results of the classifier and put together different regions of the genome, unbias them, and write a regression model which is composed of multiple sites. So this is basically what Sapir did. She trained a different model for every cell types, for every such block, every such marker. And she wanted to use this model to classify and estimate what % of cells originated from that specific cell type. So let's go back to the base theorem. And remember that, imagine that we have this pattern coming from this window here. Okay? So we would want to estimate what's the posterior of coming from a lung cell given that we've seen this pattern. Okay? And using Bayes' rule, we could basically say that this is proportional to the likelihood of this pattern from a lung cell times the prior probability of seeing a fragment of DNA from the lung. Okay? So this is the prior part, which is kind of cool because you could put lots of medical knowledge into that. Maybe this person has some earlier lung disease and so on. So this is something that the physician should like. And this is something that we should like because it's basically the likelihood of some pattern given a specific label. And you could write that using different models of different complexities. One thing you could do is you can say that we have an independent model. So the likelihood of green, red, green, green is just the multiplication of 3 greens and 1 red. Yeah? Like a binomial distribution. Or you can say that, no, we want to apply here a first-order Markov model. So basically, this one is independent, and then the following site depends on seeing a red site that preceded it, and then green given red, and so on. Or you can write it into more complex model, like a second, third order, and so on. You basically approximate the joint probability using different levels of details, complexities. So let me show you how this worked, and then we would switch gear and move to something a little different. The first thing is in silico mixing results. So once again, we mix in the computer maybe 3% of DNA from neurons, fragments from neurons, into white blood cells. Okay? And then we run the algorithm, and we get that it's, let's say, 3.4% estimated here. And we could do that multiple times. Or you could look at 1% or 0.1%. This is the range that we want to be at. Before we were at about 1% error, and for early detection you would want to be at 0.1% or below. And you can see that the models work pretty nice, but at some point they break down. So this was the independence model, the zero Markov-Odell models. But if you look at k equals 1 or 2 or 3, you see that they become slightly more accurate as you increase the complexity. And you only double the number of parameters per window, so it's not really a big issue here. Okay? So here you see neuron, fallopian or chatzotzrot. You see heart cardiomyocytes, which work beautifully, and you see liver. Okay? And you can also see it in other cell types like breast epithelial cells, or thyroid, or prostate, or pancreatic beta cells. So I'm really happy with those results. And the cool thing is, for those analyses, we just use 25 genomic regions for each cell type. So this is something that they could do, that Yuval and Ruti in Yuval Dor's lab could do easily for every cell type using 25 markers in one experiment, basically. And this yielded about 0.1% of accuracy, which is enough for early detection, which could be enough for monitoring or estimating if the treatment is working or not, and so on. Okay? So that's the applicative part of this talk. I would kind of skip the biology part, but I just want to give you the bottom line. So those markers, we now know that they fall into two categories, or we now think. It's very preliminary. Most of them are just enhancers. So enhancers are regulatory regions that are far from genes, and they fold over those genes to control their expression. So if we look at the markers that we found for different cell types, they all look like enhancers because they have the known sequences for proteins that are only present in that specific cell type. So they have enrichment for those transcription factor binding sites for the proteins that are present in those specific tissues and bind DNA. So we found those sequences in the markers. If we associate them with genes, let's say we look at the markers for heart, and then we look at the nearest genes, and we ask what is special, what is common, what those genes are enriched for, then we see highlighted in yellow terms like cardiac and heart and contractility. So basically, the markers are near genes of relevance to that specific tissue. If we look at their DNA accessibility, so we see that it's about 10 times higher. So they're much, much more open in the cell types that they regulate compared to other cell types. So this is what you see here. You see a window of 10,000 bases to the right, 10,000 bases to the left, and you see the average accessibility of those markers, and you see that they're much, much more accessible here than in other cell types.
How do you measure this accessibility?
It's a cool method that you basically throw scissors at the DNA, and then you sequence the fragments that are short enough. So if you're accessible, you would be hit by multiple scissors. Yeah, you would be cut by, and you would be shorter. So if you sequence the shorter fragments, then you must be...
And this accessibility is not just a feature of the DNA itself, but also of various methylation and other things that we regarded that relate to what's really being...
I'm not sure. I think it's correlated with those, but I'm not sure that the method itself is dependent on methylation. I imagine not.
No, but I'm asking, so the DNA accessibility, would it look the same for every cell in my body?
No. So you see that the blue, so in blue you see the accessibility. Let's say that those are heart markers, blood markers. Now we're looking at blood markers. So the accessibility in blood cells would be 10 times higher here than at the surrounding region. And it's also 10 times higher, but you don't see this enrichment in other cell types in the same regions. So it's like tissue-specific accessibility.
So the type of things that cause a certain region to be more accessible or less, so there are many factors. One,
it's all connected.
And we don't know all of them?
It's all connected and cross-connected. We know methylation is the most stable. So Chaim Seder would tell you that methylation is the most stable mark, and it underlies everything else. And it's hard to change. And then you have the packaging of DNA. And then you also have the location of DNA within the nucleus, which is what Amnon Buxbaum is studying. Because some cells, some fragments of the DNA are kind of pushed to the corner and they're being silenced just by the mere location inside the cell.
But I think Ron's question is there are layers of information. Who's writing who
Yeah.
is not trivial, but we know that the methylation is kind of self-sustained. So everything, it's not affected by other things. Mostly it's affected by other things.
And it's the hardest to change. And then you could look at the chromatin, and again you see that those markers that we identified are enriched for marks for active regulatory regions. So again, we think those are enhancers. And if you look at gene expression, like whether the genes that are next to are expressed or not, so it was really nice. I don't know much about biology, but if you look at the heart markers and you look at the nearby genes, you find NPPA and NPPB. Those are the proteins that you measure after heart attack, for instance. Those are the proteins that are used for measuring the heart functionality, together with troponin. And you find myosin heavy chain and myosin light chain, which are basically the proteins that make the heart contractility. So those are, those are the gene next to the markers, which is really cool. If you look at the pancreas, you find insulin, which is basically uptake of sugar into the cells. You find glucagon, which is the signal that the pancreas releases to release sugar into the blood. And then in the liver, you find the glucagon receptor, which is basically the other gene that listens, monitoring the levels of glucagon and release sugar when needed, and other protein, other liver protein enzymes, and so on. So without knowing much biology, I came up with those really and without like trying too hard, I found those really cool stories about the markers. Okay. And the last thing about those markers is that I think that they are working as 3D anchors that could be turned on and off. So this is super preliminary, but there's a protein called CTCF. And CTCF is basically the protein that functions as this arrow of bringing one region of the genome next to the other region of the genome. And this is one of the positions in the genome, which is unmethylated, only in the colon. So this is a colon marker. And what you see is that CTCF is bound there. Other people published that, but you see that CTCF is bound, is present in that position in all cell types except for the colon. So what we think is basically that in the colon, this position is methylated, and then CTCF cannot bind there. And so basically you break apart this connection, this 3D fold, and you alter the 3D structure of the genome. Just by it's like you have those light switches that are covered with the plastic in the mirpeset, so you could turn on and off the light. So I think that this is the same idea, that if you methylate this region, you prevent the 3D folding from happening. Okay? So this is what we think about the markers. And if you have 5 more minutes of patience, I want to talk about forensic applications, which are cool. And then I would finish. Okay? So this is Roy. And Roy is a student that took my course some years ago as a Talpion. And then he disappeared. And then he called me one day and said that he was recruited by Mazat. He is now a policeman while being a soldier for Talpiot. And they had a problem, because often they would run into a complex crime scene. So what do I mean by complex? It means that you have more than 2 people involved. So in the crime scene, they would find DNA of the victim and of one suspect and of another suspect. Okay, so they had DNA of those 3 people in the crime scene. And they also could tell, using proteins and other methods, that the DNA came from blood and sperm and urine, for instance. Okay? Yeah? So they know that those three people are involved, but maybe some of them are not guilty of a rape, maybe. Maybe just urinated there a day before. So what can they do? And we came up with this idea that basically when you sequence this piece of DNA and you measure the methylation of DNA, we capture both the methylation state, the epigenetic marks of this DNA, but we also get the DNA sequence in between them. And some people show some position in the genome are different between different people. We have about 10 million known positions in the genome. That some people have A there and some people have G, for instance. Or maybe some people have A from their mother and A from their father, and other people have A from their mother and G from their father. Okay, remember genetic course from 3rd grade. So maybe we could make use of that to associate basically what tissue came from which of the three persons. And Roy built this new human forensic methylation atlas from published data. So we basically, here you have 800 methylation sites now, and you can see that some are methylated in the blood, but not in the skin or the sperm or urine, and some are specific to the skin, and some are specific to the sperm, and so on, okay? So now you could imagine this scenario. Imagine this row here, okay? So what we see here is here. We see a genomic position, and we actually measured that. Ruti and Yuval measured that in their lab. So what we see here, and maybe I should give credit to Daniel Sher too, right? Yeah, okay. So what we see here is a genomic region that is fully methylated in the blood, fully methylated in the urine, but is unmethylated in the sperm. Okay, so if we see an unmethylated molecule, then it must be from the sperm. But then we could also look at nearby positions, right immediate to this methylation site. There would be a position that differs between different individuals, and maybe the victim has A on 2 of their DNA copies there, and one of the suspects also have A on 2 copies of DNA, the mothers and the fathers in this position. But the other suspect is heterozygous, so they have both G and A. You see where I'm getting at? And we have 10 millions of those sites. So clearly you would find a position that is different between suspect B and the rest, or suspect A or the rest, and so on. So we basically learn decision trees that they say something like that. And this is from data that we already measured in the lab, in their lab. So the idea is like this. You take a piece of DNA and you ask, is this read or those 3 sites methylated or not? If they're methylated, then they could be from any of the blood, skin, urine, and I don't care, I don't want to look at them anymore. But if they're unmethylated, then they must have originated from the sperm. In which case, we can look at an adjacent position on the same piece of DNA and ask whether it's G or A or C or T. But if it's G, then it must have, given that the world is composed only of those 3 cell types and only of those 3 individuals, then if it is G, then it must have originated from suspect B. So basically, and indeed we see that at those molecules that we sequence, we see that 99.5% of them came from the sperm of suspect of that person. Okay, we did like an experiment in the lab. So this trajectory would implicate the sperm to suspect B. Got it?
Beyond a reasonable doubt?
So the thing is, you don't need to be beyond the reasonable doubt here, because you don't need to select this person out of the entire universe. You just need to, because he was already... Yeah, you need to rule out that this DNA, this sperm came from the 2 other, because you already found DNA of that person using the standard method of DNA, they use Bdikat Abaut, which is like repeats in the DNA. So they already found his DNA in the crime scene. Now you just want to associate the sperm to him. So this is something that we think that could work. Anyway, so that was the talk. Great thanks to Yuval and Ruti and Ben. that all the parts of the talk are in collaboration with them, and they do all the molecular analysis, and Ben is doing all the clinical work, and I'm just doing the computational stuff. Josh was a MD PhD student of mine and of Yuval, and he's graduating next week, I think. Nathaniel and Sapir, Nathaniel is a PhD student, Sapir was a master's student. They both will receive tomorrow their master. And Roy that I talked about the forensic part. And then we have some people that are giving us DNA, and of course the physician and people in Yuval's lab that did the actual work. So that's it. Thank you. So if you're not tired, we could take questions, or we can break and I can answer privately, whatever you want, in Hebrew, whatever you want.
Okay, so you tell me.
Bye.